Students, today we will study Oedogonium. It belongs to the class Chlorophyceae, order Oedogoniales, and the genus Oedogoniaceae, family Oedogoniaceae, and the genus is Oedogonium. Occurrence: the Oedogonium is a fresh filamentous algae with more than 400 species, usually present in the permanent water bodies of water such as pond, lakes, and shallow tanks. The filaments are attached to the rock, log, etc., or are epiphyte on the aquatic plant and other algae. Some species are Oedogonium terrestris, are terrestrial, growing on the moist soil. The genus is represented in India by more than 200 species, the common one being Oedogonium cardiacum, Oedogonium aster, Oedogonium elegans, etc. Thallus structure. The thallus of Oedogonium is multicellular and filamentous. The filaments are unbranched and uniseriate. All cells of the filament except the basal and the apical one are cylindrical and alike. The basal cell, which act as a holdfast, is devoid of chloroplast. Its proximal end is extended into the finer finger-like or disc-like projection which help in attachment to the substratum. The terminal or the apical end is rounded, elongated or acuminate. Thus, the filament shows clear apical basal polarity. Cell structure. The cell has a fairly thick rigid cell wall which is differentiated into an outer chitin, a middle pectin, and an inner cellulose layer. The center of the cell is occupied by large vacuoles. The protoplasts form a thin layer in between the central vacuole and the cellulose layer. The chloroplast is reticulated, extended in the form of a sheet throughout the protoplast. Most of the stand of chloroplasts are parallel to the long axis of the cell. Many pyrenoids are present at the intersection of the chloroplast reticulum. The cells are uninucleated. The nucleus lies in the middle region of the cell and in between the chloroplast reticulum. A characteristic feature of Oedogonium is the presence of distinctive transverse band at the distal end of some cell. The band formed at the time of cell division is called apical cap, and the cell with the apical cap is called cap cells. Growth and cell division. In Oedogonium, growth takes place as a result of division in the intercalary cell. All the cells of the filament except the holdfast and the apical cell are capable to divide, but divisions are confined only to few cells. The cell division in Oedogonium is a complete process. The important event during the cell divisions are: the cell division is initiated by the formation of ring of wall material in the upper part of the cell, which gradually increases in thickness. The nucleus migrates to the center of the cell and divides mitotically. During the telophase, an unbranched floating septum is laid down between the two daughter nuclei. The septum is formed due to a continuous accumulation of vesicular material, microtubules, etc., from the cytoplasm. The ring of the wall material now elongate, causing a split in the parent wall near the apical ring. In this process, a part of the ruptured parent wall remains attached to the anterior end of the new daughter cell in the form of a cap. It is known as the apical cap, and the cell having the cap is called the cap cell. The other portion of the ruptured parent wall can be seen at the proximal end of the new cell. The floating septum gradually migrate upward to the base of the daughter cell and develop into a mature cross wall. Thus, each cell division of the cell results in the formation of an apical cap. The number of apical cap in a cell indicate the number of the time it has divided. Thank you.